this meeting, so I'll just have to try and cope. This is the kind of introduction slide. Let me see. Which shows you the principal reaction which the enzyme acetylcholinesterase performs. Hydrolysis of acetylcholine, acetylcholine. Acetylcholine is a neurotransmitter, both in the periphery at the neuromuscular junction, in ganglia and in brain. This enzyme is very rapid. The turnover time of 250 microseconds, which is needed for its biological function. And this speed is what's called diffusion control, which means if you put the enzyme in a assay vessel and you add glycerol and increase the viscosity, the enzyme slows down because acetylcholine can no longer reach the active site of the enzyme fast enough. And this assay, its principal role is rapid termination transmission at cholinergic synapses. But we'll have some reservations about that in a few minutes. It's of interest to toxicologists, pharmacologists, clinicians. It's a target of carbamide and organophosphate insecticides and antiparasite agents, of organophosphate nerve agents, and of the first generation of drugs for treatment of neurological diseases, myasthenia gravis, glaucoma, Lambert-Eaton syndrome, Parkinson's disease, and Alzheimer's disease, where it's so far, except for one glutamate antagonist, it's the only, the anticholinergics are the only drugs for treatment of the disease, though one should have no illusions. They do very little, they just slow down or increase your cognitive power at the very early stages of the disease, and then they stop working because the treatment is symptomatic, not therapeutic. This is a picture I like to show. I actually showed to the neurologicals in Woods Hall. It says there, it's called pseudocholinesterase in the brain. It's by a very eminent neurobiologist at MIT called Anne Graybill. It was published in Nature 82. Pseudocholinesterase is the old name for butyrylcholinesterase. And this shows the lateral geniculate, which is a kind of halfway station from the eye to the visual cortex. And it has alternate bars and stripes of acetylcholinesterase and butyrylcholinesterase. And when you make a lesion and denervate laterally, you see changes in the intensities of the bars, the layers of these two enzymes. And this is published in Nature, even though nobody knows what it means. Usually, I don't know with physicists, when biologists or biochemists send a paper to a journal, they say, oh, this is very nice, but what is the biological significance? So in this paper, there's not one word about biological significance, and the situation is still the same. This is 1982. This is almost 40 years later, and the situation is the same. So now, I said the main function is usually believed to be termination of transmission of cholinergic synapses, but we have what are called non-classical functions of AChEs, whether real or conjectured. So A, we have acetylcholine as a substrate, but not as a neurotransmitter. For example, what you might call a trophic factor, that it in some way changes innervation or denervation or some other functional entity in the nervous system. B, this is very much what Conrad will say, AChE is a protein generator. It hydrolyzes acetylcholine very rapidly and can produce very large pH changes locally. 3, action on one or more other substrates. For acetylcholinesterase, so far there is not one, but for butyrylcholinesterase, I'll show you in a moment that there is. Finally, acts as adhesion molecule, and I'll go through these things quickly. So, non-classical functions. You see here, here's a paper, the first one by Paul Lair, who only recently last fall closed his lab in Darmstadt, very serious neuroanatomist and developmental biologist. This is a paper in 1983. Comparative localization of acetylcholinesterase and pseudocholinesterase, or butyrylcholinesterase, during morphogenesis of the chicken brain. And what he shows that in the chicken brain, before there are any synapses whatsoever, you have very tightly regulated projection of acetylcholinesterase and butyrylcholinesterase in cells, which make very exact and precise migrations. We have no idea what these enzymes are doing biologically whatsoever. Secondly, mirobrazin, this is even early 1966. localization of AChE in axial membranes of frog nerve fibers. You find it both in the myelated and non-myelated regions. Again, we have no clear idea what it's doing. 3. The paper by Susan Greenfield and collaborators in 1985. A non-cholinergic function for AChE in the substantia nigra. This is a place in the brain associated with Parkinson's disease, which is neither cholinergic or chemoreceptive. Neither receives cholinergic inputs or generates ones. But there's lots of acetylcholinesterase there. What it's doing, we do not know. Next, osteoblast-derived AChE, a novel mediator of cell matrix interactions in bone. And Paul Geneva gives quite convincing circumstantial evidence that the AChE is an adhesion protein associated with phosphatidylinositol adhesion in bone. Then there's a review by Carl Silman, and finally, a very exciting paper published just over 4 years ago. Plasma-butyrylcholinesterase regulates ghrelin to control aggression. I won't show the biochemistry or the pharmacology, but ghrelin is I think about 30 residue oligopeptide, polypeptide, with an octonyl 8-carbon residue at the end, and it's involved in aggression and in appetite. And Brimjoy and his colleagues were working on knockout mice, noticed that mice which had the butyrylcholinesterase knocked out, the males, were very aggressive and could not be caged together. And then they went into this further, they found that the butyrylcholinesterase actually hydrolyzes ghrelin, and this controls aggression. So this is the first clear evidence for a clear biological function of butyrylcholinesterase, which until then was mostly hand-waving. And here I show you the fact that AChE may be an adhesion molecule is not perhaps in retrospect surprising, because there are 4 cholinesterase-like adhesion molecules known, which are very closely homologous to AChE and BChE in their structure. 3, glautactin, glutactin, and neurotactin are from Drosophila, and 1, which is probably now the most famous, is the neuroligin, which is famous now because you see this reference by Bourgeron et al. Is associated, if you have autism, you have autism-related mutations of neuroligin and also mental deficient mutations. So this is now a very, very popular topic for neurobiologists to study, the involvement of neuroligin in synaptogenesis and connections in the brain. And the last reference there to Yves Bourne and Pascal Marceau, they have the structure of neuroligin and it's complex with, they never say it's been not neurotactin, anyhow. Let me just show you the next slide. This just shows a putative structure of neuroligin in complex with neurexin, and below you see acetylcholinesterase. You see how similar their structures are. So the likelihood that acetylcholinesterase is an adhesion protein is very high, and there's a very simple smoking gun experiment. If you knock out the active site residues, and it still acts, it must be not catalytic. And although I believe that Carl Silman has done this experiment for acetylcholinesterase in the connection of bone, he's not published it yet. So the smoking gun is still there, and it has to be looked into further. Here I show a picture from a review by my friend and colleague who died, unfortunately, about 7 or 8 years ago, Jean Massoulier. As you can see, he liked ice cream very much. And this shows many, many different forms of acetylcholinesterase, and the same is true for butyrylcholinesterase. And in terms of molecular biology, they are generated mostly by alternate splicing at the C-terminus, and followed by post-translational modification. And I draw your attention here to 2 of these forms. We have these collagen tail forms here, where 3 subunit tetramers are linked to a collagen tail. And then we have, up here on the right, we have tetramers which are linked to hydrophobic peptide, transmembrane peptide. This is the principal form in skeletal muscle. This is you always find this form in the cholinergic synapses in the brain. There's also a dimer here. Which one is it? Too many forms here. There is a dimer which we'll come back to later, which is, it's here down at the bottom, which is GP anchored. It's linked by each subunit of the dimer has two phospholipid molecules anchored covalently at the C-terminus, when they anchor the enzyme in the plasma membrane. We'll come back to that later. Now, let's just go back. These forms, these tetramers, three tetramers of the collagen tail, and the one with the transmembrane polypeptide, use an anchoring device which is not used by any other protein as far as we can know, has ever been found. It's specially designed to anchor acetylcholinesterase correctly, either in the periphery or in the CNS. And the way it does this is shown in the next slide. What Weizmann did in terms of molecular biology was to dissect out by removing and monitoring the effect of various sequences. He found that the sequence responsible for anchoring the acetylcholinesterase subunit to the membrane is an amphiphilic helix at the C-terminus, about 40 residues. And he then found there was the collagen tail, which I showed you before, has a sequence which is rich in proline, which interacts with these subunits. And what we did with Jean many years ago, in 2004 in fact, and Christine Garbet, was to synthesize these two sequences, the proline-rich attachment domain called the PRAD, and the WAT, which means the amphiphilic attachment domain, which contains three conserved tryptophans. They were synthesized by Christine Garbet at the School of Pharmacy in Paris. And when they were mixed 4-to-1, we got the complex that you see on the left here. This is a very, very tight complex in which you have a superhelical coil around the polyproline 2 helix, which goes in the opposite direction. And the coil itself is a structure which was solved by Francis Crick about 70 years ago. He also saw the polyproline 2 structure. He saw both these structures apart from dealing in DNA. And but what is unique is that this is the only structure in which you have a coiled coil or a superhelical coil around the polyproline. You often have coiled coils in isolation. This is the only structure in cholinesterase which contains this structural feature. And this is a model which we generated and published in 2004. And we are in the process of working on acetylcholinesterase now. But butyrylcholinesterase has this similar structural feature. And we together with our former student, Sevilla Silman, who is now at Utrecht, she used cryo-EM to solve the structure of the butyrylcholinesterase tetramer. You see here the 4 subunits arranged around this superhelical structure with a polyproline going through it. In this case, the polyproline is not a special collagen as is found in the neuromuscular junction. The butyrylcholinesterase recruits lamellipodin to help itself assemble into a tight tetramer. And we're now, hopefully, in the next few weeks or so, we'll have the same structure for acetylcholinesterase. And this shows, just to fill in the picture, some EM pictures of the acetylcholinesterase. We have pictures like this, but these are actually, I think those, I'm sure, actually from Jean Massoulier and his colleagues. And this is a more schematic on the right, a more schematic version of what the structure looks like. And in the brain, you have the same tetramer assembly module with the superhelix and the polyproline, but the attachment is to a transmembrane polypeptide. I say this arrangement, the assembly mode, is unique to acetylbutyrylcholinesterase. And then on the right, I show a slide from work I did when I was on a sabbatical in London with Eric Barnard in the late 1970s, and we developed a protease cocktail to prevent acetylcholinesterase being chewed up when you homogenize muscle or other tissues. And then we did sucrose gradient centrifugation, which means you layer the enzyme sample, the extract, on top of a sucrose gradient, and then you spin it onto a centrifuge for many, many hours. And you count in Swedberg units how far the protein migrates down the gradient. And the bigger it is, the faster, the further it migrates. And what you see at the top is the posterior latissimus dorsi of the chicken, which is a fast twitch muscle, and it has exclusively, or not almost exclusively, this A12 form, which is apparently the functional form of the synapse. Related, actually, macrodissected and pooled and analysed similarly, single-end plates. In the anterior latissimus dorsi, which is a slow tonic muscle, you have almost only this dimer. In cardiac muscle, you have a tetramer. And these forms are presumably chosen and selected to anchor the enzyme in the right distribution, the right orientation, the right density in the synapses. The level of detailed understanding we don't, this has still to be deciphered. Now I'm going to switch gears a bit, and we're going to talk about microenvironmental effects on enzymic activity. And I'm going to mention three effects: diffusional effects, static effects, and the lipid environment. And these are two slides, one from work which was part of the PhD thesis of Rachel Goldman and involved Roy Kaplan, some of you may know, or Academy and Frank Kachalski, which was done in the year before I left from my postdoc after I finished my PhD, which was on insoluble enzymes, enzymes coupled to insoluble polymers. In this case, when we talk about the insoluble membrane, we actually soaked papain, a proteolytic enzyme, into a collodion membrane and cross-linked it so it would not leak out again. And then we studied its behavior. And what this curve shows is, we look at the hydrolysis of an ester, and the ester generates protons when it's hydrolyzed by the enzyme. And this curve is normalized. What it's showing is that, whereas in the case of the soluble enzyme, you get a bell-shaped curve, as is common for many enzymes, in the case of the membrane-bound enzyme, you get first as you get increasing, you get a kind of plateau, and then you go up again. And this curve is normalized, so actually this curve should be down here and going like this. And what is happening is that as the pH goes up, the enzyme acts faster. The protons released don't diffuse away fast enough from the enzyme to prevent the local pH decreasing, so you get negative feedback. And 2 years later, I was working with Acetylcholine at Columbia as a postdoc, and I was looking at a membrane, a purified membrane fraction from electric eel, electrophorus electricus, enriched in acetylcholinesterase. And I was doing that mostly because I was still waiting for some electrophysiological equipment to arrive so I could start working on the electrophorus and do electrophysiology on the single cell preparation. And what we found was very similar behavior. That if you look at the enzyme, this is under a pH stat where you measure the activity by maintaining a constant pH by adding alkali from a syringe to neutralize the protons produced by hydrolysis of the acetylcholine. And what you see when you do this is if you do this, normally you're expecting a bell-shaped curve. What we got was actually, as you see here, we got a sigmoid curve like this one. What we realized was that it was the same phenomenon. In this case, it was in a macroscopic scale with a very thick collodion membrane. On the right, with acetylcholinesterase, this was a plasma membrane. We were working with it in a pH stat without buffer. And because there was no buffer, the local pH at the membrane could be affected by this rapid hydrolysis of the acetylcholine by the enzyme. If you just added a smidgen of buffer, 2 or 3 millimolar tris, we could completely eliminate the effect, and we'd get a bell-shaped curve, just like we do if we did another assay, a cholinergic assay of the enzyme in solution. This work was not done by me, but by actually Leon Goldstein, one of my teachers at the university, with Frank Kachalski, at about the same time, a little earlier back. This is the effect of electric field on enzymic activity. And what Leon and Hugo Levine did was to embed the enzyme that working with trypsin inside a polyanionic matrix. And they looked at how this affected the activity. And they found two phenomena. First of all, the substrate, which was a homolog of arginine, because it was positive, was dragged into the matrix and this increased the Km. The enzyme, the substrate was bound much more tightly. And the other thing they found was that the pH profile was also changed because the salt was drawing protons into the matrix. You had to raise the outside pH much higher than you would expect for the native enzyme in order to get the same pH curve. And both these effects, you can see these graphs, these tables here, both these effects were almost completely reversed by ionic strength, which screened the electrostatic field of the polymer and brought the enzyme more up the situation it was in the solution without the polyanionic matrix. So this is a field effect. Now, I mentioned before in the slide I showed from the review of Jean Massoulier, we have an enzyme form of acetylcholinesterase, which is a hydrophobic dimer. You find this on the surface of red blood cells. In other places, you find on the presynaptic membrane of the synapse in the electric organ of Torpedo californica. And if you actually homogenize the tissue and do sucrose gradients, you find that 2/3 of the enzyme is this dimer I'm showing you here, and 1/3 is the collagen tail form I mentioned before. The collagen tail form is found in the basal lamina between the presynaptic and postsynaptic membranes. This enzyme is out on the presynaptic membranes. What its function is, we don't know. You can perhaps conjecture that since the electric organ tissue contains is so active and releases so much acetylcholine and has to produce recurring shocks very rapidly, perhaps it's necessary, in addition to the collagen tail enzyme, to mop up the excess acetylcholine, but this is just pure hand waving. And on the left you see my former student, Tony Futterman, who's now a professor at the Weizmann. We collaborate on other projects. It's only the opening pitch for the Boston Red Sox. I won't explain why. And on the right is a torpedo electric, a torpedo fish that we use. We use Torpedo californica usually, which we get frozen tissue in California. And maybe I'll go back. Oh, sorry. So this enzyme is a dimer with a disulfide link in the two subunits, and it has a hexasaccharide extension at the C-terminus, which is added post-translationally. At the end of this chain, on each subunit is one PI molecule, which is covalently attached to the oligosaccharide, and the two fatty acids chains of this PI molecule, 4 for the dimer, are what anchors the enzyme in the phospholipid bilayer. When you solubilize the enzyme with a bacterial phospholipase, which was developed by Martin Lowe, our colleague, the enzyme becomes totally water soluble. And this for us was actually an important experimental tool, because this was the enzyme which we crystallized and solved the 3D structure of a few years later. But in this case, we're talking about the enzyme in which the two PI molecules are still retained, and with this we could use to anchor the enzyme, this was together with Matthias and with Conrad and their students, to anchor the enzyme to a monolayer, as you see in the next slide. This is a lamellar trough, which you see or have seen in Matthias' students' presentations. We have a lamellar trough here, and we introduced the enzyme on the lowest, from below, from the medium into the monolayer, so it faces outwards. And then we come onto its action with a light source and a photodiode built into the lamellar trough for this purpose. This just shows, if we get the detergent-soluble AChE, how it's incorporated into the monolayer as measured by lateral pressure. And then these two slides you actually saw from Matthias yesterday, so I can go over them very rapidly. They just show that the peak of acetylcholinesterase activity coincides completely with compressibility very, very well. In the next slide, I recommend just show this one. You know, I didn't, yeah, it was saving slides. So this shows that this effect that you observe of the coincidence of compressibility and of the peak of activity are independent of the lipid used, of the temperature, or of the pH. It's a phenomenon which is related to the compressibility of the monolayer. And this is a kind of schematic explanation of why this is, that near the compressibility maximum, you have a very variable modulation of states, fluctuations, and it's these fluctuations which indirectly accelerate acetylcholinestase activity in the enzyme in the monolayer. And now, these are two references which, again, may have been seen from the work of Matthias' student Weizmann, from Bernhard Fichtel and Shamit, who is here, who showed that you could get, you could initiate a wave in a monolayer by applying a proton impulse within the Prof. And together with Bernard, this work was extended by incorporating acetylcholinestase into the monolayer, just like before, and looking at the propagation of acid excitation, which you see on the right, going towards pressure sensors. And here is a light detection apparatus, just like we had before, to see what's happening to acetylcholinesterase in the course of the movement of the pulse through the monolayer. What you see here is that you have an initial rate of acetylcholinesterase activity, and when the pulse moves through the monolayer, the compaction of the monolayer causes a raise in local pH, which activates the enzyme. And as the pulse goes through, the activity appears to stop, and then resumes its original levels. Here you see initial rate, enhanced when the pulse reaches the enzyme, then turned off, and then resumes the original rate. In controls, you see, without any enzyme, you see no effect of the pulse. And we attempt to explain this in the terms of the graph of the work with acetylcholine, which I showed you previously, that initially this compression leads to a higher the pH and to activation of the enzyme. This produces protons which turn off the activity by feedback, and then after the pulse has passed, eventually you go back to the original state. So these are the three phases. The initial phase, the phase of increased activity caused by compression, the inhibition, the feedback inhibition caused by proton release, and finally back to the initial stage again. And Matthias suggested this actually could be a method of signal transmission within a cell, which would not be diffusion control. We could actually get a wave like this, which could transfer a signal from one protein or one protein complex to another within the bilayers. This would be a very rapid signaling mechanism, much, much faster than the diffusion of second messages from one complex to another. No, I still have some time, right? How much do I have? 5 minutes. 5 minutes, okay. 5? Okay. This is a slide which defines what those conformational diseases or proteopathies. And these terms, they both refer to classes of disease in which certain proteins become structurally abnormal and disrupt the function of the cells and tissues and organs. And often the proteins fail to fall into their normal configuration, and they become toxic in some way, either gain a toxic function or lose their normal function. This includes Alzheimer's disease, and what we call other amyloid diseases, after the amyloid A-beta protein. Parkinson's disease, synuclein. Creutzfeldt-Jakob disease is a prion diseases, mad cow disease, and other prion diseases. And here you see some of these plaques within the brain of an Alzheimer's patient. And with my colleague Weizmann, he and I have spent a lot of time looking at the dimeric form of acetylcholinesterase is a model for partially unfolded protein because when you put the protein into mild denaturing conditions, it produces a partially unfolded dimer, which is very stable over long periods of time and allows to do a lot of physical chemistry on it. But I'm not going to talk about that. And here you see this conversion of an enzyme from a native to a molten globule to a fully unfolded state. It's interesting, I was a protein chemist since my PhD started in 1960. At that time, for the next 20 so years, all the very great theoreticians in the field of protein chemistry, Silman, Silman, Hanford, Sharaga, they always assumed that when the protein got off the ribosome, it remained fully unfolded and, until it eventually found its right configuration to fold correctly. It was only in 1990 or 1991 that all they realized that when a protein comes off the ribosome, it immediately collapses to a molten globule due to the hydrophobic interactions, in addition to the charge interactions, and from the molten globule it shifts to the native state. And again, we used for this purpose, for our studies, the same dimer hydrolyzed by the phospholipase to become fully soluble. And what I show in this slide is the effect of DMPC liposomes on thermal inactivation of torpedo AChE. And torpedo AChE is, in the absence of a liposome, is stable at 28 degrees. But in the presence of a liposome, the enzyme inactivation is greatly lowered by interaction of the enzyme with the lipid surface, and it rapidly inactivates. You can see the open circles with the liposomes compared to the closed circles in their absence. You see the enzyme activation is greatly decreased by this interaction with the liposome. And then you will also find if you do a sucrose gradient in which the liposomes, which are very light, float to the top of the gradient, the enzyme which is bound to them floats up with them. So it's unfolded by the enzyme, by the liposome, and attaches to it. And the next slide shows there's a peptide. What you can do then is take this complex, digested trypsin, and the enzyme is completely digested except for that part, which is bound to the liposome, which you can then solve as a detergent and send to be sequenced. And we found this particular sequence, this green sequence, which interacts with the liposome. And we also modeled how it might lie parallel to the bilayer. And there are known proteins which have actually been solved in which the membrane-binding region is actually not inserted into the bilayer, but lies along it. So that's a plausible possibility. And the last two slides are from a collaboration with Cassian Bon, who many years ago was a student of Jean-Pierre Changeux, and is unfortunately no longer with us. And we showed the same effect of liposomes on thermal inactivation of the snake venom enzyme, Dumer's AChE, except at a considerably higher temperature. This is at 45 degrees, rather than 28 degrees. And finally, the last slide, we show that in fact a kinetic process. You can describe it just like an enzymatic reaction. The enzyme in this case is a liposome, and you can get through to Km and a Kcat. On the left you see what's called a Michaelis-Menten plot, on the right you see the line with a bird plot. So the interaction of the DMPC liposome with the enzyme is actually a situation in which the liposome is the catalyst and the enzyme is being unfolded. And that's my last slide. Thank you.
Thank you very much. We have a question. I'm going to have to pass the microphone. While we ask questions, there's a second speaker to come and get set up. That would be great.
Thank you, Israel, for the talk. I have a question. In the middle of your talk, you were referring to these monolayer experiments, and this word of compression arises, but in the previous and after descriptions of measurements of acetylcholine in biological systems, you don't see this compression thing. Can you maybe comment on whether these measurements in monolayers and compression are related to the biological systems, or are these two completely parallel mechanisms?
I have no biological system on which I work or I worked on where this might happen. I would think that people like yourself and Christian might perhaps know the answer to such a question better than I would. I just think that the explanation in this case is reasonable. And we do know that you can take a cell which is surrounded by a bilayer, you can quite extensively, quite greatly extend it. The bilayer does not fall apart, it can stretch a lot. So it could happen, but I don't know biological cases.
I mean, it's probably unlikely to assume that there are no state changes in the biological system. And the compression modulus is just a measure for the state, I would say. So,
I would like to make a very brief comment, because it will not be taken up in the roundtables, not anywhere else, about esterase and catalysis and how it relates to this product. Just very, very briefly, because it does not take up. The point is that in these measurements with the monolayers and the esterases and other enzymes, there is the second law of thermodynamics proven to be by exactly the same law as underlies the quanta of Planck and Einstein, or exactly as the same law underlying the action potentials as we see it. The cause and the origin from the second law of thermodynamics locally of catalysis in itself. And just to outline the point, because it nowhere appears anywhere else, it's one identical law, one law underlying, it's a law of Planck-Einstein for the quanta, it's a law of our monolayer gains with the action potential, and it is a law of the origin of catalysis, which may be much more important than anything else we discuss if we look at the importance of catalysis. And the point is that it proves the compressibility is just the same sign we see with the Langmuir arrangement. It's indeed the second derivative of entropy, which means it's the least squares of Gauss. It's a very fundamental thing, and this is a fluctuation, the uncertainty of quanta. It's the uncertainty of the protons in the motion. And so compressibility is just a way to see it with the Langmuir trough. It's indeed, and anywhere in biology where we don't deny the validity of the second law of thermodynamics, this must happen. And the way how catalysis comes about is exactly how Einstein derives diffusion to come about. He also takes the second derivatives, the fluctuations, and then you take an average and you have your clock next to it and you get fixed second law. And exactly the same thing happens here, that the fluctuations of protons are very, very strong. And these fluctuations are by the Einstein relation for diffusion now related to the reaction rate. So the rates are much faster because the origin of proton diffusion is very large, and with the proton diffusion the reaction goes forth and back. So this is the physical origin of catalysis, which we will not come back in this session, but it is in a way, in the long run, and of course with all the chemical industry and the patents for enzymes, much more maybe important than anything we discuss about this action potential.
If you promise to give it to 30 seconds, because we're already running like that. All right. Thanks. Well, that makes a good transition with